The last presenter today is Dr. Brian Christie. Dr. Brian Christie obtained his PhD at the University of Otago with Dr. Cliff Abraham. He then received postdoctoral training with Dr. Daniel Johnson at Baylor College of Medicine, and then with Dr. Terry Sichnowski at the Salk Institute as a Howard Hughes Fellow. Dr. Christie then accepted his first faculty position at the University of South Dakota School of Medicine in 1999 and was subsequently recruited to UBC in 2001. In 2007, Dr. Christie moved his laboratory to start a research program at the newly created UBC Island Medical Program in Victoria. And as part of this move, his primary appointment was transferred to the University of Victoria, where he founded UVic's neuroscience graduate program in 2009, serving as the inaugural program director and graduate advisor until 2018. Dr. Christie's research program focuses on brain plasticity with a particular emphasis on promoting functional recovery in both acquired and congenital neuropathological conditions in the aging brain.
All right, thank you for that kind introduction, and Randy, thanks for the invitation. As is, maybe evident from that my background, resilience plays a big role in all of our research, and we do a lot of neurodevelopmental work. We work with Fragile X and FASD, prenatal cannabis, psilocybin exposure, and incorporate that with a lot of synaptic physiology. I've got the little joke about we also study brain injury because I've got the nice big cast on here, which really is brain injury, which I managed to avoid during that tumble down the stairs. Joking aside, this talk's really going to involve some subtext resilience components, and a lot of it's going to involve what the trainees went through, even what the PI goes through as it. And so I encourage you to think about that in the question session at the end, and maybe feel free to ask. So I'm going to tell you a little story about traumatic brain injury, and it's really how do we look at this. And for those of you who aren't familiar with traumatic brain injury, there's a lot of different levels of it. You can have very mild subconcussive blows, and people are engaged in sports like hockey or football and stuff like that. In a typical practice, they're going to have 10 to 14 subconcussive events during the course of a practice. That may or may not result in a subconcussive blow that transitions into a mild traumatic brain injury, which can be no loss of consciousness to any loss of consciousness under 30 minutes. And this is the injury I'm really interested in, this mild traumatic brain injury versus the more moderate and severe traumatic brain injuries, which usually involve severe damage to the brain and more likely to be fatal. So the CDC has given us this model, typically of the coup contre coup injury. And I think everyone is familiar with this type of schematic, and the whole idea being that there is some sort of injury in the front of the brain, some sort of injury in the back of the brain, and that leads to this breakdown of the brain. You're going to have ionic fluxes, blood-brain barrier breakdown, apoptotic processes, inflammation, excitotoxicity. And how do we study this? Well, ideally, we'd want to use humans, but we have a very low application rate of individuals wanting us to whack them over the head with a hammer, and the ethics department tends to get on you if you try and have individuals participate in. We did manage to do one study where we had soccer players who were at a soccer academy engaged in heading practice, and we were allowed to measure their cognitive performance before and after just their one heading soccer practice. So we weren't actually doing the manipulation, and in fact showed that there was a decrease in cognitive performance in these athletes at high school levels after a heading practice. So that would be our ideal subject if we can do things. But typically you end up with post-mortem or post-injury individuals, which all have their own unique challenges, including the availability and their ability to stick with the study. You could use in silico models, but really they're primarily for studying the physical forces involved in TBI incidents, and they don't yield a lot of accurate structural and functional data. So we end up going to animal models. And so I'm going to start off talking about the development of animal models and how we went about it. When we entered the field, there were really two main models for head injury. There's the fluid percussion injury model and the controlled cortical impact model. Both of these are invasive. They involve craniotomies to open access to the brain, which causes inflammation on its own. For the craniotomy, you have to use anesthesia, which is neuroprotective in itself, and it can also cause changes in body temperature, which are associated with tau hyperphosphorylation. This altogether is going to help promote inflammation in the brain. And then finally, to be at the end, it involves directly impacting the cortex. It's not really a concussion, and the brains don't look anything like we would see in an MRI or CT of someone who's just had a concussion. This would be severe damage to the brain. We wouldn't expect a lot of functional recovery from this tissue area. Most mild traumatic brain injuries recover quite well. So we started looking at what was going on, and surprisingly, Canada is really good at this. And if you look in the top left-hand corner of the slide, you can see that these closed head injury models have popped up with quite a lot of regularity in Canada. So we have this awake closed head injury model I'm going to show you today that we developed in our laboratory and was taken down to Australia by Dr. Sandy Schultz. But there's also the pressure wave model from Ted Allison's group at the University of Alberta, the ladder impact model that was pioneered by Rochelle Maciasiak that we've also started using in our laboratory. It's quite good. And then there's Cheryl Wellington's Chimera model. And they're all different ways of doing a closed head impact like normally happens with concussions without directly impacting the brain. Now, when we started developing our model, I was just talking to Brianne, and it was a first at the University of Victoria to do a brain injury model. And so the student in the top right, Alicia McConey, ended up going through a year-long process to get through animal ethics, prove to them that we actually weren't hurting them all. And in fact, if I showed you a video of the animals, they pop right out of these little plastic cones that we use to restrain them so that they're held in position. They have a little helmet on their head that helps distribute the force of the blowover, and we don't actually hit them. We actually rapidly accelerate their heads with a CCI piston, but with a little rubber grommet at the bottom, so it helps distribute the blow, but also gives this rapid movement, kind of like what you'd get in a car accident. Now, with the CCI, normally, when you're penetrating and striking the brain, doesn't matter how hard you do it, whether it's the mild on the far left or the severe injury on the far right, you get a lot of brain damage. And this is what your typical brain looks like post-perfusion. Even the perfusions are difficult because you've damaged a lot of vasculature in the brain. In contrast with the awake closed head injury, it's really hard to tell if you've got an injury. The brain's perfused really nicely, and even if you do crestal sections through the middle, you don't see a lot of necrotic death. So we have a brain that you can actually look at in the first time and go, where's the injury? Well, that ends up being a question that we pondered. And instead of having this coup contre coup injury model, where you have the top and the bottom of the brain being injured, we started thinking about a diffuse center injury model, or the center of gravity model, as it's been called. And here, when you're moving the head in any direction, it's really the center of mass of the brain that's going to be moving around. And it's going to shift and pull and create shearing forces around the surrounding tissue. And now you'd start to expect to see those injury patterns in the middle of the brain rather than at the top of the bottom. So now we actually have a testable way of looking at brain injury. And so this is what Katie Neal, one of the first graduate students to get involved in this, started doing when we actually were able to use the model. And she did a very simple study where she actually did some BRDU staining to look and see if neurogenesis was impacted by this injury. And kind of like with epilepsy, you'd expect a big proliferation of neurogenesis. And those neurogenic regions are what are highlighted in the little circles on the top of the brain in a control animal. And you can see they're much larger in the injury model. And I'm just going to blow that up a little bit so you can see it. And you can see that there are black dots everywhere. I'm not sure, I'm not going to try and touch all the buttons on this to try and get the little light working, but you can see there's little BRDU positive cells everywhere outside of those neurogenic niches that we normally expect to see them in the hippocampal dentate gyrus, right? It's going through the CA1 region, down into the thalamus, up into the lower portions of the cortex. Definitely not the pattern that you expect with that coup contre coup injury model. It's more the center of mass model. And so what's causing all these things? They aren't just all new neurons. And in fact, it turns out that they're going to be microglia. So I'll shed that little bit of the story. And on this study, and sort of recapitulating what Brian showed us earlier, we have those normal surveilling microglia. You can see these activated microglia, and there's a little bit of purple in the middle of the screen, which is actually fibrinogen. And fibrinogen is a blood product that's released when you have microvascular damage. And so you can see these glia stretching up towards it. What was interesting to us is we also saw these rod microglia in our brains. And these are microglia that are associated normally with Alzheimer's disease. So really unusual microglia formations in our brains. We also saw phagocytic microglia, right? So ones that are starting to encompass the fibrinogen of the brain. And I'll show this a little bit better in the next slide, but I wanted to get across that we had this distributed pattern of these microvascular incidents with our wake closed head injury model. So we didn't see lots of gross necrotic cell death, but we could pick up microvascular damage. And here's a great example that a PhD student, Juan Trevino Paredes, picked out, where he actually found a broken blood vessel in the thalamus, showed the leaked fibrinogen that was coming out around it, and then the microglia that were starting to move in towards and encapsulate the fibrinogen. And this is within 24 hours of the injury. 1 hour post the last hit. So you can see this is a direct, rapid movement. And this is going to be below the resolution of anything you're going to see with an MRI, right? We're going to be far below 50 microns. This is going to be sub 2 microns. And we see these amoeboid microglia macrophages interacting with the leaked fibrinogen. So now we have something that's drawing in microglia to the site, causing them to be activated. And there is, again, the ramified microglia. And what that does is it gives us a little bit more of a model for what's going on. So if we have these mechanical forces engaged that are causing microvascular damage, these are going to activate microglia. And fibrinogen and fibrin can actually bind with that CD11B interaction site on the microglia and lead to the release of cytokines, IL-1 beta, TNF alpha, CD86, et cetera. And so now we have a method for causing increased demyelination, neurodegeneration, and possibly those persistent cognitive and emotional deficits that are associated with mild traumatic brain injury. And these activated microglia could result in excessive pruning. For the sake of time, I removed the spine story because I didn't want to go overboard. So now we have targets, though. And so now we can start moving from mice to men. Does anybody here ever read the book of Mice and Men? Thank you. I appreciate your education. No one in my lab got that picture. They didn't know who John Steinbeck was. So, so we engaged in started transferring this to the clinical realm, and this work was taken up by a new PhD student, Taylor Snowden, who started a set of studies, and then the pandemic hit. And so during the pandemic, she was able to use that time to reflect, develop her computational capacity, and to look at what was going on in the realm of MTBI. And one of the things she found in her summary review was that individuals who had mild traumatic brain injury, not severe traumatic brain injury, but just mild, were twice as likely to be developing or diagnosed with dementia and AD later on in life. That's still a significant rate. It actually just slightly beats out obesity as a risk factor. And it's about half that is severe TBI. So if you have a really severe brain injury, you're more likely to have dementia and Alzheimer's disease later on in life, almost 4 times likely. And of course, the APOE4 that Dr. Wellington is also very famous for studying is your leading candidate for Alzheimer's dementia later in life. But it gives us some targets that we can start looking at. We know that these individuals may be more likely to develop Alzheimer's disease. So what do? Well, Taylor said, let's study the elderly. And so she had to show a lot of resilience. So she first came up with her idea, and it's like, well, we really need to understand how badly damaged any individual in our study's brain is. So she decided she needed to learn how to do MRIs and MRI analysis. And for this, we are lucky that we had Dr. Jody Gowrilick on campus, who specializes in doing MRI with elderly patients. She also wanted to do biological sampling for our laboratory, and that was to do blood and saliva markers. And of course, as a brand new PhD student, she has none of these skills. So we sent her over to VIHA, and she learned how to be a phlebotomist, and now she's an excellent phlebotomist with H patients, so she can get blood and saliva samples from them. And then she also learned how to administer cognitive tests, which were then going to be evaluated by Dr. Gallerluck, who's also a clinical psychologist. And finally, being a near Olympic athlete, she really wanted to get exercise into any of her studies in some way. So she wanted to involve aerobic exercise to see if that could help remediate any of the damage and improve cognitive function in the elderly. And she also started working on a program called NeuroTracker that it gives the opportunity to do cognitive training. So because of all the delays of the pandemic and getting the ethics re-established, and then collecting patients who are going to be interested, or clients who are going to be interested in partaking in the study, we've just gotten this going, and we just have a few preliminary results. The first one that popped out, even with the first 10 clients, if you had under 3 concussions, you weren't as likely to have severe symptoms as individuals who had had 4 concussions over the course of their lifespan. And these are all individuals who are over 55 years of age. So over their lifespan, greater than 4 concussions, 80% of those subjects indicated that they experienced regular headaches. So there's a little bit of an indication that there may be something going on. Now, the cognitive assessments, we actually turn around and get analyzed by a certified psychometrist. So we're still waiting for that data to come back, and Taylor is anxiously awaiting that data and phoning the psychometrist daily almost to see how it's going. But we're going to have a lot of things to look at: global cognitive function, memory, executive functions, and then some concussion-specific analyses, including the SCAT5 and King Devitt and VOMS. That'll give us an idea of both their executive cognitive functioning as well as some of their vestibular function. I promised that I would get in a picture of a U-Haul truck for Nick. Yeah, and I also snuck in, it's got magic mushrooms on this side. Yeah, inside joke, sorry for that. Because I was using it to hide this little MRI scan showing some differences in the brains that we've already seen. Again, this is in the first 10 subjects, and we're looking at individuals who have greater than 3 concussions in their brain. And when I start the movie, what it's going to show, you're going to see these little red areas, and they're going to be in the centers of the brain, just like we saw in those mouse models. And this is areas where basically the water molecules aren't flowing unidirectionally. They're flowing in distributed directions, which is a measure of fractional anisotropy. I think I have that right. Did I get a nod? I got a nod. And so it indicates areas where there may be breakdown in the structure of the brain. So let's see where they are. I think there's a little delay. And so if you keep your eyes focused towards just off the midline, you'll see a little bit of red flash up. There we go on the right-hand brain. On the middle brain, it'll be towards the top, around the edges of the corpus callosum. And in the left-hand side, I don't think we have anything showing up in that one because it's just the midline scan. So we have some evidence that we actually have damage in the middle of the brain, kind of following that center of gravity hypothesis in individuals who had mild traumatic brain injury sometime in their past. And of course, all the data looking at exactly how long ago that was is still to come. But this really, setting us up well, and it helps direct where we want to go with our fluid biomarker analysis. So we're kind of going to start focusing on these general neurodegenerative and aging associated biomarkers first, including the general inflammation ones, IL-1, TNF-alpha, et cetera. I saw some excellent posters in the back that have the same sort of mindset, looking for dementia-related things. But there's a whole host of things out there. And so this is one of, sort of the openings that hopefully are associated with this resilience series, is that we have blood, saliva samples, MRI scans, cognitive assessment data, and we're always open to looking for partners who may want to bring their expertise and develop a line of inquiry with us. With that, making close to my 15-minute timeline. I'd like to thank everyone for attending, and also, of course, thank the funding as well as our collaborators, Dr. Jody Garlick, Dr. Sandy Schultz, who has just moved from Monash University, one of the top institutes in Australia, to Vancouver Island University. His wife and family all live on the island, and so he chose joining them over living in Australia. And he's just getting a research program ongoing there. Just won a Michael Smith Award as a scholar, and also an NFRF award. So he's getting things off to a good start. And he's collaborating with Dr. Wellington and UBC and myself at the University of Victoria. Great person to get to know. And of course, all of the trainees who, without them, none of this would be happening. So thank you.
Hi, my name is Shakiva from SFU. So I just had a clarification question for the mild TBI that was like a predictor of dementia. Is that just one like concussion, or is this mild like multiple, or yeah?
Yeah, so for that group that I showed today, we did 8 repetitions of this awake close head injury. And some of the data that I pulled out just to shorten the talk is looking at whether you can give that over 1 day, over 2 days, 4 days, 8 days, etc. And what we see is that the pattern of post-neurological assessment doesn't vary depending upon the number of injuries that you receive, even if it's in 1 day. So you'll end up with some sort of deficit. So you might score on a neurological assessment protocol, let's say a 9 out of 12. But on the 8th one, you're still going to be around there a 9 or 8 out of 12. It isn't a cumulative injury. And that's how I first thought it might be a cumulative injury. So we get more injury the more we gave, but they seem to be like 8 distinct events. And you may have multiple, microvascular incidents in that brain, but they don't necessarily have to be in the same place. Yeah, so it is multiple.
Hey, Brian.
Hey, Nick. Oh, yeah, that was all for you.
Nice talk. Got me thinking about a couple things. So one, just curious, how long after the mild TBI model do you get BBB permeabilization? Like if you have any insights into how long that lasts.
Yeah.
And then second, the shear stress just made me think a lot about mechanosensitive channels and how they might be playing a role in microglial activation or changes. Just wondering if you had any insights on this.
No, and if you know anyone who is interested in mechanosensitive, receptors on microglia, I don't know anyone out there in the audience. No, yeah, no, we'd be more than happy. We've got tons of samples and that. Yeah, the permeabilization piece has been difficult. Like we look for just breakdown of blood-brain barrier, and we tried a number of different ways. Evans blue and polysaccharides, all kinds of tracking things, and none of them really satisfied me as for the veracity of the approach. And I also became more and more aware of the distributed differences in permeability in different parts of the brain, right? And it's more permeable in the areas where we're more likely to see damage. And so I think that just noticeable difference is a lot harder to detect there. Nevertheless, we see that significant microglia proliferation around that region that says something different's going on, right? And it might not all be the fibrinogen, but our next tack would be to look at ways in which we could block fibrinogen activation of microglia, and also following sort of Brian's lead, looking at ways that we could reduce the oxidative load and maybe even changes in glycolysis, stuff like that could accompany these types of injuries. Yeah.
Right.
Hey.
Let's talk. Two questions. The exercise program. Has that been worked out yet? What you're actually going to try and do? It's because these are probably new dwelling out in the I guess. But.
Yeah, and so there's a mile exercise program, sort of walking, and we talked about aerobics with Taylor, and she's a training for triathlons and actually Ironman right now. So none of them want to train with her, understandably. But you know, Victoria, and she lives out in Sydney. So she works in Victoria, lives in Sydney, which are two heavily, you know, populated areas with, you know, people 55 plus. And we've also looked at Zoom. So there's in-person and Zoom training. And right now it's been a Zoom training session with them doing sort of mild exercises to get their heart rate up for a certain periodicity and then back down. And they're doing that for a 12-week. And also, you know, we've given them Fitbits so we can look at the amount of activity they have in walking and stuff like that.
You do agree with compliance in that context?
For, you know, for this group, yeah. And I think it's because it's the first wave, right? So it's the first 100 tend to be the enthusiastic individuals who want to take part in these things. And there is quite a large community of that in Victoria. You know, the newly wed, nearly dead. Yeah. You know, sort of, you know, does hold true there. And we have a lot of, you know, well-educated, you know, individuals, you know, ex-doctors, nurses, psychologists, et cetera, who are really engaged and want to take part. So we've had high compliance so far. Yeah, what's that?
They're highly motivated individuals.
Very highly, yeah, yeah.
Second question is, you made a very good case for thinking about the nature of closed head injury, congenital injury, this for the central thing.
Yeah.
Is there possibly there could be a bit of both?
Yeah, I mean, yeah, if you think about age and, you know, the firmness of suture lines, I think the younger you go, there may be more flexibility in the skull, which could allow for more of that pressure injury on the outsides. But our you know, skulls and the meninges and the, you know, pressurized vascular system are there for a reason, is to provide a really significant protection from your head, your brain rattling around your head like a pea in a pod, right? It just doesn't operate that way. And so that you know, coup contre coup motion of the brain inside isn't really there, but that center of mass really is. And there's some researchers down at the University of California, San Francisco, who have, you know, started modeling it in more detail, looking at the differences in density from, you know, structures like the thalamus and having the corpus callosum there, and the pull you can get from that, along with the pressure waves along the vascular or the cerebrovascular system, right? So all of that comes into play, I think, with those types of injuries.
Thank you, Brian, for that great talk, and thank you for, I know my trainees really felt when you were talking about the ethics challenges of being the first lab doing dramatic brain injury at a university. So I know my trainees appreciated your mention of that. Two questions. When you were looking at the VRDU, did you scan for neurons? So that I know that was microglia. Did you look? Was there any effect on neuronal neurogenesis? And my second question is about the saliva that you're collecting in the older adults.
Mm-hmm.
Do you know what you're going to look for in those saliva? Because you mentioned a bunch of different markers, but I was just wondering if you know, because we might be collecting saliva from older adults for circadian measurements, and I'm just wondering if there are other things we should be looking for in the saliva.
I mean, we've so on the second question first, we've always had an interest in trophic factors, and because we're going to have a group that's interested in these, you know, physical interventions and also cognitive interventions, we suspect that we should be able to pick up increased levels of brain-derived neurotrophic factor and, you know, stuff like that in those individuals. Yeah, the you know, it's, you know, ideally, you know, we'd sample in here. We're still waiting for ethics approval on that. It would be the first, yeah. You know, so we kind of look for multiple ways that we can do it. The blood, you know, it can be, wow, we got a bird visiting us in the back. Sucker, I'm out of here. You know, so you know, you can have BDNF production in the testes and all kinds of different places that are completely unrelated to what's going on in the head. So we're hoping that by multiple measures we'll be able to hone it down a little bit more. But we have some direction, but we have the capacity to fish as well. So that was, and what was the first story again? So, I mean, we have done co-labeling studies. So we look at the age and state of the neurons, and that's still ongoing. So we've done an aging model with the RMTBI work, and that's why we haven't published anything out yet. And so we've got young animals, sort of pre-puberty, early adulthood, and late adulthood. The late adulthood, the aged animals are all done, and we didn't see a change in neurogenesis after MTBI, but we did see the proliferation. And so we did triple labeling on those two, and they all, come up as glial subtypes. In fact, we see astrocytes and microglia in those. So we're, looking to continue that work and look to see if we can find evidence that these are now becoming, cytokine positive cells. I think a lot of the strategies that, Brian's pioneered with his work are where we're going to end up following that. It's always, time, money, and post-pandemic, right? The pandemic really played havoc with a lot of the research plans that we had. Yeah.
Hi, Brian.
Hey.
Hi, over here. I have a couple of questions for you on Zoom.
Sure.
The first is from Janine Martin, who would like to know how you determined how many concussions the participant had. Was it based on medical data or self-report?
So we ended up having to use self-report, but we do document where they have medical support for their concussions. We have in our assessment guidelines sort of inclusion-exclusion criteria for what would be considered a concussion. Recognizing that this is all retrospective data, right? Your ability to remember those concussions might be impeded by having those concussions. And we preceded this with some other studies. One was called the Good Old Days study, where we just looked at how well they remembered that past life and what sort of reflections they had. And people tend to, divide into either negative, emotionally charged memories or positively emotionally charged memories. And it's surprising how many people flip all these events over into the positively charged, the golden years, the good old days, right? High school heroes, that kind of stuff. We all hated high school when we're in it. Doesn't matter how long ago it was.
Thank you. Bryce Phillips wants to know about a couple of slides mentioning glutathione and oxidative stress. Do multiple MTBIs throughout the lifespan lead to increased susceptibility to oxidative stress and neurodegeneration?
So can I say this conclusively? No, but yes. We did a really nice proteomic scan with a proteomic center in UVic and did this MALDI looking at lipid profiles and saw dramatic changes in lipid profiles, particularly glutathione in the brain following concussions with this ACHI model. So that gives us, a little bit more impetus to, start following up these microglia roots and looking at how they could be regulating the injury. There's also work from Rochelle Machiesiak's laboratory showing that the glymphatic system is impacted, right? So that's that sleep-wake cycle and clearance. So there's multiple angles that glia can be affected. And then we've been following up on synaptic physiology. So we're looking at transmitter release, AMPA-NMDA receptor ratios, the capacity to induce different types of synaptic plasticity, whether it's NMDA dependent or endocannabinoid dependent plasticity. And we see changes following repeated traumatic brain injury. My poor postdoc is back in Victoria right now doing this painstaking study of giving one TBI and then doing synaptic plasticity on the animals, following the timelines that we use for the multiple injury thing. And it looks like with our model, at least the awake close-hit injury model, we don't see a significant effect, which is why we brought in the lateral impact model from Rochelle Machiesiak, because one TBI on those seems to have an effect, and a repeated injury on that gives you a more significant effect. And I suspect with the Chimera model, you'd see something very similar. Yeah, thank you.
Quick question. This is a great talk, and I really liked your graded model for TBI. Seems like, well, a big difference from the severe to mild, it's also damage to the meninges. You mentioned meninges. We're gonna kind of interested in that too.
Yeah.
I feel like this is a transition that meninges is damaged. Do you have any observations or thought on that?
So, yeah, and in fact, in our younger animals, where we had more flexible suture lines, sometimes we'd see damage to, some of the cerebrovasculature and the meninges associated with pressure and stuff like that. Those animals were more likely, one, to show an acute seizure activity, which would subside, and they tend to clear the meninges tend to clear out any blood and stuff like that, and the pressure fairly quickly. Rodents are fairly robust animal models to use, but with the animals, they seem to get over it really quickly, and so there's a different timeline there than what I suspect you'd see with humans. And a damaged meninge, you're already into moderate severe diagnosis already, right? So
Thanks, Tom. Wanted to speak to this in a general way. I wondered about comparing and contrasting expression of injury in concussive style injuries versus PTSD.
Yeah. So we've got a protocol in to look at PTSD that I'm still waiting to be signed off on. Resilience, that's PI resilience there. But I do think that, all right, here's a great example for the weight close head injury. I actually thought that going back in that bag and going through this injury profile might induce a PTSD-like event or anxiety in these animals. And we never saw that. They would happily go back into the bag, and it's almost like they're snug and safe in the little tube. There were others that first time they went in, they'd be squeaking. 8th time they went in, they'd be squeaking. Like they didn't want to be in a closed environment. But we didn't see differences in, open, field. We did, Y maze routines to look at, alternating, behavior seeking. We did see some issues with motor behavior that seemed to persist a little bit more, and that might be related to vestibular function with that center of mass type of injury. But for the PTSD, it seems that you have to up the force of the impact. So rather than this accelerating the head movement that we did with the weight close head injury, the lateral impact, where you're literally firing a piston into a helmet on the side of the animal's head. So it's like being hit from the side by a car. There we see more anxiety, more deficits in synaptic plasticity. So that's that graded injury approach that I think Brian was looking at. And those animals are actually, I had a meeting with my group this morning, and they were just starting the IBA1 stain for that group. So we'll know more. This summer is going to be really exciting. And we're going to have lots of tissue, lots of blood samples and stuff like that. So if people are interested and have questions that they want to pursue, we're more than happy to ship things off. It's I think getting the most use out of the samples that we have is the best way to move forward, right? And we'll also be putting our data up online for if people want to look at our electrophysiology or whatever. We're with that new data elements package that CIHR has brought in, we're trying to get ahead of that wave and put things up so that everyone can look at it, because you never know what you might notice that I miss, and those are interesting, insights.
Hi.
Hey,
Just sort of a clarification with the micro injury sites with the vasculature, do those heal eventually? And if so, are they more likely to be future injury sites if there's repeated incidents? Sort of.
Yeah, it's funny. So my idea, and especially like the model I drew up there, would be that one injury site might be more prone to having multiple injuries. And that we actually never really saw that. Sometimes we saw a little bit larger vasculature breaking, but because we also did like 8, it was hard to tell if the first or the 8th did it. So that's another study that my poor postdocs had to do, where he's done 1 injury a day and then has to sacrifice, and then done 1 injury for 2 days and then sacrifice to try and look for that. And he didn't see in his preliminary look an increase in the incidence of microvascular bleeds across time. So it didn't look like having one made much. And in fact, his insight was that he thought that having one, you were more likely to damage weak spots. And then later on, you would have fewer because you'd already damaged the weak spots. So, you know, it's hard to say, you know, we obviously don't have power to really say anything significantly, but it was enough for us to go, okay, this isn't the most worthwhile low-hanging fruit to pursue right now.
I have a similar question about the relationship between MTBI and PTSD. So prior to starting at UBC, I worked at the VA hospital in Boston, working with some vets who experienced combat-based blows to the head repeatedly. And in some of our work, we found that even though there was loss of consciousness for some of these blast bomb events, they had really salient traumatic memories. And so, you know, MTBI and PTSD are so highly comorbid. So I don't know if it's a question or a comment, but I know that the PTSD affects some of the white matter tracts that are affected in TBI, and the importance of trying to separate the effects of the two, the psychological and the physical. And so, yeah, I would, you know, be interested in chatting more with you about that and thinking about the best way to sort of do that. And one thing that I think is important in humans, and I'm thinking specifically about intimate partner violence, which is associated with repeated mild traumatic brain injuries, like the locus of control and the unpredictability of when the next one might come has enormous psychological tolls on that could exacerbate the physical impact of the trauma. So yeah, just would love to chat more with you about that topic.
Yeah, no, and that's a great thought line. And I really actually am invested in that. And I actually learned something really new. So here's some, you know, trainee feedback I received. I was interested in using the SCAR model of intimate partner violence and pairing that with TBI. And I was really excited about this and telling one of my female PhD students about this, how this would be such a great model to use. And we could start looking at, you know, the comorbidity between intimate partner violence, where there's often choking, head movement, and then head injury. So there's lots of things, anoxia and damage going on. And she was kind of quiet and she went away for a day, and then she came back and said, "Can I talk to you?" And I said, "Sure." And she goes, "I don't think I can do that. I, you know, find that so uncomfortable and, you know, feel so, you know, awful. And it just trying to engage in that type of research that I just don't think I can do that. Do you think like I'm not going to be a good scientist because of that?" And I'm like, no, everyone has their moral boundaries and you should try and stick with them. So it's something I learned. I never, you know, thought about who I was talking to when I was doing that. And I know I talked with Paul van Donkelaar, and he was getting sort of the same feedback when he was going to CIHR, like, why is a man leading this charge for women's intimate partner violence? Well, why not, you know, is one answer. But he responded really well, and he brought on a team of females to lead a lot of the interactions, because if you do have PTSD, the last person you want interviewing you is going to be a guy, right? So there's lots of different things that you can learn from following these question lines and looking at it. But I honestly think that the PTSD model needs to be explored a lot more, because the mechanism of injury is a lot more complicated than anything that we see with just the TBI or with just the SCAR model, right? The you know, conditions here, whether it's anoxia or you know, being more prone to inflammation and stuff like that, are going to change the outcomes of the brain injury that they encompass. Yeah, happy to chat about that anytime.
So I think we'll try to wrap things up here. And so I'd like to thank Dr. Brian Christie for a wonderful talk and engaging.
Thank you.